Okay, so VSV multiplication cycle. This is the model virus that we use to learn about rhabdoviruses, which are negative single-stranded RNA bullet-shaped viruses. I think, if I remember correctly, they were about 75 nanometers in diameter and 100 nanometers in length. Okay, so let me get my brush. There we go, and red ink. Okay, so you can see here's the bullet-shaped. Here's a oh my gosh. Here's the bullet-shaped virion, and you attach using LDL receptors. So LDL receptor. If you notice, it uses the flat side of the bullet, and this is, I don't think this was ever stated explicitly why, but Dr. Samuel did say that VSV or rhabdoviruses attach preferentially to the basal or the flat side of a monoculture of cells. And looking at this picture now, I would venture a guess that it's because it uses the flat side of its bullet, so it would, with the flat side of its bullet, it would want to attach to the flat side of a cell. It's not really shown here with this curved drawing that this curvy cell that Dr. Samuel has, but imagine monolayer where you have semicircular cells. Anyways, you have attachment, and then you have receptor. I'm just reading it off right here. Receptor mediated endocytosis. So it's a bit different from poliovirus, where, I mean, it's similar in the sense that they both have to, I guess, get absorbed into a cell, or the cell brings it in, and they both have to attach to receptors, obviously. But remember, in the polio one, some viruses, they can just simply inject their genome into the cell. But this rhabdovirus, it actually has to go. There's no second options. It has to be engulfed by the cell via receptor-mediated endocytosis. So now that's in here. Excuse me. Now that's in here, it has fusion. So fusion is just basically fusion is just basically when the virus, with its viral envelope, the viral envelope becomes part of the cell. It just becomes one with the cell, I guess, because when it's still in this endosome, or basically a virus inside a vesicle, it's really not part of the cell yet. It's still very much, there's a wall between the virus and the cell. But with the fusion, finally, it's almost like you have viral genome just swimming around in the cell, as if it was a part of the cell. Okay. So you have fusion, and then you uncoat the genome, right? Because if you can't uncoat your genome, what's the whole point? I always bring up this example. You have a crazy zombie virus, and you can't uncoat the genome, and the genome can't do its thing. Your zombie virus is done. It can't do anything. Now, with the uncoating, this was the first virus that we learned about a concept called virion-associated polymerases. And basically that means that in addition to capsid proteins that are, that come with rhabdovirus, embedded in that are polymerases. So you can see here, there's this L. L is I don't know why they chose L, but that is the abbreviation for the polymerase. This P is also called NS protein. P is, so NS stands for non-structural protein. The P specific, and then when you use the other name of NS, the P stands for phosphoprotein. phosphoprotein is important because it, in addition with your L polymerase, is able to, it helps the L polymerase do its thing. The L polymerase needs the P to do its thing. And then you also have your N protein, you know, quote unquote N right there, these little tiny orange circles. These are your nucleocapsid protein. So that's the stuff that the native ssRNA genome, parental genome, is attached to. So in addition with the, again, with the structural proteins of rhabdovirus, the structural proteins, by the way, are called the matrix proteins, abbreviated M, as in mommy, M as in mommy. In addition to those, you have the L, the P, and the N coming with the virus. And specifically, the L and the P are virion-associated polymerases. So you have this native ssRNA genome. So unlike polio or Picorna viridae, where Picorna viridae can immediately start the first round of translation, the basal level of translation, VSV or rhabdoviruses can't do that. And because they can't, do that because the native ssRNA is not an mRNA. So the first thing it has to do is transcribe or replicate or convert its native ssRNA into a positive form. Now the only problem is that cells, you know, our cells, don't have any sort of polymerase that can read native ssRNA and make protein out of it. And this is why the virus needs the L polymerase and a P phosphoprotein to come with the virus. It's because the L polymerase is the only thing that can make proteins after oh no, sorry. Okay, so I messed up a bit. Our cells don't have the ability to read native ssRNA and make positive ssRNA. We don't have that kind of polymerase. We only have DNA polymerase to replicate genomes and RNA, and we only have RNA polymerase too to make the mRNA from DNA, but we have nothing to read native ssRNA and turn that into positive ssRNA. So that's why the virus needs the L polymerase attached with it. It's because with the L polymerase can read the native ssRNA and convert that into positive ssRNA. And once there's a positive ssRNA, then the virus can hijack our ribosomes and make protein. So you can see here, you have your so two things happen. The L polymerase makes a positive single-stranded RNA molecule right here. What it also does is that it transcribes. Now, the way that rhabdovirus controls what proportions of its proteins to make, like what to make in the right amounts, is that it has 5 open reading frames, and each reading frame are separated by intergenic regions and polyuridine sites. So when you have the viral polymerase transcribing positive, or transcribing essentially messenger RNAs, sometimes it can, when it, you know, another thing about the genome of rhabdovirus is that it only has one start site, right? One start site controls one start site, which begins at N, the structural protein, it controls all the expression of all this. So you have one start site, but then you have multiple 1, 2, 3, 4, 5 intergenic regions, 5 polyU sites. That is a perfect setup for transcription control. And the way that the transcription control works is that when you have the viral polymerase coming down, it will always start at the N, and it transcribes all the N perfectly, and then it hits the poly-U site that in the intergenic sequence that's right before the P. Now, most of the time when it hits that U site, it goes to a thing called attenuation, which, if you call attenuation, is basically the polymerase slips and stutters on a U and then it falls off, right? If it falls off, it can't transcribe the P. The P doesn't have its own start site, so now that polymerase has to, the P gene isn't transcribed. If it wants to be transcribed, then the polymerase has to go back to beginning where to the N is, and it starts again. And then second time it goes through, it's like, oh, poly U site slips and stutters, it falls off again, and does that again, and then it falls off. And maybe sometimes it's lucky and it's able to transcribe the N, and if, ooh, yay, fine, gets the P, transcribes the P perfectly, and then gets to the M, and the same thing happens. There's attenuation at the poly U site of the right before the matrix protein gene, and then slips and stutters and falls. You just repeat this over and over again. Once in a while you're lucky enough to get the N, the P, the M, the L, and the G genes transcribed, and all that together allows you to transcribe and express the genes that are required in high amounts. So those are N, P, M, and G. Specifically, N is needed in the highest amount. So that's why it's the first gene to be it's the one. N is the gene that's next to the start site. And you get high levels of N, but you don't really need high levels of the L polymerase. The L polymerase is an enzyme. You only need a few, and you can do a lot with it. So, to wrap up what I was saying, you have the L polymerase. Okay, it replicates the positive. It makes a positive ssRNA, and then does the primary transcription using the same L polymerase. From here, you'd find how translation of our proteins, so translation C, N, P, M, and L. Now that you have these proteins, you can now make the basal levels of the positive ssRNA right here. You can now take those and replicate a bunch of native ssRNA genome in a short period of time, right? So you have all this genome, okay, all that genome. And down here, this pathway right here is only for the G protein. Basically, the G protein needs to undergo maturation in the endoplasmic reticulum and the Golgi apparatus. So then you have your G and your N structural proteins here. You have a bunch of your genome here, essentially, and then the structural proteins and the virus receptors get embedded into the cell membrane. So that's right here. And then you attach your matrix protein to basically the carboxy terminus of your virion, antigens. And then that attracts the countless amounts of the countless amounts of genome that you have. And then finally you start to have budding, and then you get a bullet-shaped virion. So the bullet-shaped virion is actually due to the way it buds, because it, I guess, protrudes out the antigens first, and then puts and then attaches on the genome as it's budding out. It buds out with a bullet shape. One thing that I forgot to cover, the secondary transcription. Basically, this is saying with even higher amounts, after your first round translation, with even higher amounts of your L polymerase, you can now take these negative ssRNA that you made at the same time, and then transcribe and translate those, and you essentially have like a, I'm gonna use blue. You essentially have this repeating. You essentially have this repeating cycle of making more protein, making more genome, making more the positive ssRNA to make even more native ssRNA to make even more mRNA messenger RNA transcripts to make even more protein. Yada yada yada. So this is Rhabdovirus multiplication cycle. Now time to move on to measles or Paramyxoviridae.